would like to introduce you all with our next chair, Dr. Rajesh Sharma, and our keynote speaker, Dr. C. Kartikeyan, to our esteemed audience. I would like to invite on the stage Dr. Rajesh Sharma and Dr. C. Kartikeyan. Dr. Rajesh Sharma had done his MBA from DAVV Indore. He had done his PhD in pharmacy from DAVV Indore. He had done his M.Pharm from DAVV Indore. He had done his B.Pharm from DAVV Indore. He has a total research, industrial and teaching experience of more than 25 years. He has published a total of 144 research papers published in referred journals. He has a total of 8 research papers published in non-referred and reputed journals. He has a total of 5 review articles. He has presented a total of 12 presented in conferences, seminar, workshops, symposia, and he had trained number of 13 PhD students and awarded, and number of M.Pharm students, research project supervision 60. Now I would request Dr. Rajesh Sharma to throw light on the topic for and invite Dr. C. Kartikeyan.
Very good afternoon to all. Namaskar. First of all, I would like to express my sincere thanks to my teacher, the advisor of this conference, Professor Piyush Trivedi sir. I also would like to express my sincere thanks to the principal of this college, Dr. Rizwan Khan, Dr. Shikha Agarwal, and whole organizing committee. I am happy to share that today we have a very renowned speaker, Dr. C. Kartikeyan. Dr. C. Kartikeyan is going to deliver his lecture on designing natural products like analogs for anti-cancer drug discovery, insights from nature. I would like to say we do have a glorious past. We do have more than 5,000 years old Indian system of medicine. So, Dr. Kartikeyan is going to correlate how Indian system of medicine can be used to identify a lead molecule from the herbs and plant. So, I am happy to introduce Dr. Kartikeyan. Dr. Kartikeyan is the Assistant Professor in Department of Pharmacy, Indira Gandhi National Tribal University, Amarkand. He has done his M.Pharm in Medicinal and Pharmaceutical Chemistry from Department of Pharmacy, Shri Govindaram Saxena Institute of Science and Technology. He has completed his PhD under the supervision of Dr. Professor Piyush Trivedi sir from Rajiv Gandhi Technical University, Bhopal. And I am happy to share he has synthesized more than 100 new molecules in his PhD work. He is recipient of Innovation Award for discovery of anti-cancer molecule at 3rd National Madhya Pradesh Education Summit. He has worked on a small molecule drug discovery for cancer, neurodegeneration, and diabetes. He has published so many good quality papers on drug discovery, QSAR, and synthesis. With these words, I invite Dr. Kartikeyan to deliver his lecture on designing natural product like analogs for anti-cancer drug discovery, insights from nature. Dr. Kartikeyan, please.
Thank you, Professor Sharma sir, for your generous introduction. It is always a pleasure to meet you in Indore. And let me first take this opportunity to thank my mentor, Professor Piyush Trivedi sir, to give me an opportunity to present my research, our research, in present of a very august and eminent audience. So the topic of my talk today is designing natural product like analogs for anti-cancer drug discovery, insights from nature. So these drug molecules, which I'm going to discuss today, are inspired from nature. Now, why do we need to go for novel molecules? And what is the need? Because to present the scenario, the current drug discovery program run by pharmaceutical companies is in a crisis. And why is it so? Because almost 90% of the drugs which are taken to clinical trial fails in the clinical trials, and some at a very late stage. And it takes almost $2 billion to develop a drug from the bench to the market. And if a drug fails in the later stage, then the company has already invested so much money into it, it may even ruin the company. Now, this is the latest article published in 2022, which speaks about 90% failures in clinical trials. Okay. Now, my area of focus, therapeutic focus, is anti-cancer drug discovery. The picture is even more gloomier in case of anti-cancer drug discovery, because the rate of attrition is around 3 to 5%, which means that out of the 100 drugs which are taken to the clinic, only 3 of them gets the FDA approval. And most of the times, the reason for failure is lack of efficacy, which means that in a clinical setting, the drug fails. It may be either in the phase 2 or in the phase 3. Okay, so that's why. And why is it so? Because of the reductionist approach. Now, what do you mean by a reductionist approach? Because what we do right now in modern drug discovery process is to find a target and implicate everything on the target. So one target to cure a disease. This won't work for a complex disease like cancer, where there are multiple signaling pathways, and it is a robust system which gets a cell in a survival mode. It always finds a way to survive. And that's why there are a lot of clinical failures. Okay, so what else can be done? So instead of finding one target, why not adopt polypharmacological approach? Why not go for a multi-targeted drug discovery? Okay, so that is the way forward. Now, what is multi-targeted drug discovery? Try to find more than one target or 2 or 3 targets, even many targets. Okay, and in case if in a robust network, if one system is inhibited, the other gets amplified. So if you are able to inhibit both the systems, then you may be able to achieve the intended therapeutic response. Okay, so that's what we are looking for. But it is not an easy process. Why is it so? Because it is difficult to find those targets. And this diagram which I am showing now is showing that almost most of the drugs which got approved from 2015 to 2017 are multi-target drugs. Even though they might have been, the intention might to find out as a selective drug, but most of the drugs which are getting clinical approval are working on more than one targets. Okay, so which means that this multi-targeted drug discovery has a chance, or it works. Okay, now what are all the challenges in multi-targeted drug discovery? Identifying target combinations. You know, there are multiple signaling pathways at work there. Now you have to just sort out through those signaling pathways, and you have to find out 2 nodes in which 2 nodes means like where there is a junction where multiple pathways will synapse, and then you have to find out that target and then try to find drug for that. Okay, and then the current methodology, which is more biased to the single target drug discovery. Okay, and other than that, if you are finding 2 targets, to optimize a single drug against 2 targets will be a bigger problem. Okay, so that's why it's quite a challenging process to find out multi-targeted drugs. Okay, now how to go about it? What can we do? Is there any other way by which we can find multi-targeted drugs? Why not? If nothing works, then look to God. Now, natural products which are designed naturally, okay, their there are some multi-targeted agents available in the natural products. For example, this paper talks about development of multi-targeted cancer therapeutics from nature. Okay, for example, indole, 3-carbinol, curcumin, genistein, resveratrol. Now, all their multi-targeted drugs, they were developed by the nature. Okay, and they give an idea. They give an idea to how to go about developing multi-targeted drugs. Okay, now look at those molecules. They are very simple structures. They are not very complex. And if you look at, they are structurally rigid and also they are flexible. It has both the properties. Okay, why not use this design? Why not use this design in our work to find out new drugs? Okay, now let us see what are all the advantages of naturally derived multi-targeted agents, especially in anti-cancer drug discovery. They are supposed to show broad spectrum selective anti-cancer properties. Broad spectrum, it can act on multiple pathways, so it can be taken up, it can be exploited therapeutically for multiple cancers. And another thing is, they show selectivity. Since it is not biased or it is not randomly shutting down one target, it's acting upon multiple targets. If you have seen the movie Gulliver's Travels, where multiple drops were able to pull down Gulliver, that's the way that multi-targeted agents work. Okay, now, and they have the ability to bind and modulate multiple targets. And since they are not focused or biased towards a single pathway, they can overcome multi-drug resistance. Because if any single pathway is inhibited, then there may be bypass mechanisms which will trigger the development of resistance. And another thing is, most of these drugs use the conventional way of programmed cell death for cytotoxicity. And if you look at the structure as a chemist, what I am seeing here, it has hydrogen bond donor acceptor combinations. You just look at the structures. For example, curcumin, it has a hydrogen bond donor acceptor in the middle and in the end. So this is what I see from this structure. And you can see in almost all the natural products, they have this hydrogen bond donor acceptor combinations in tandem. And why is it so? Because this is how a phytochemical has evolved along with the biological system to interact. They are designed in such a way that they can interact with multiple targets, multiple proteins. Okay, so if you just look at the naturally derived multi-targeted agents or natural products itself, you can, if you want to capture the information from a natural product, what are all the things which is present in the natural product? Now these are the most common rings which are found in the natural products. For example, quinoline, chromen, anthraquinone, indole, imidazole. So all these rings, and these are the rings which are decorated by multiple functionalities. And what are those functionalities? Hydroxyl group, methoxy group, or even an alkenone group, and also acid group, ester group. These are the groups selected by nature. Selected by nature to put on these rings, on this scaffold. Okay, why not try to emulate it? Why not to try to incorporate this design into our molecules? Okay, and another thing which is very, very interesting in terms of nature, nature is always repetitive. You see these molecules, flavones, isoflavones, the scaffold is same. The scaffold is conserved. Why? Because it has been optimized through the evolutionary process. This has been optimized for interactions with some networks. It has been optimized for in a biological system. Okay, and all the changes are in the arrangement of the functionalities in the scaffold. Okay, so why not capture this information? Why not use this information? Now, if you talk about natural products, now if these natural products are multi-targeted, and if they have all these attractive properties like selective cytotoxicity, and it has selectivity, okay, but natural products suffer from one big problem, which might spell doom to its drug development. Development of the natural product as a drug. poor ADME profile. For example, curcumin, which is the poster drug. If you take any signaling pathway, you will find curcumin acting on that pathway. You take any protein, you will find it will act in inflammation, it will act in cancer, it will act in all the diseases. But do you know that the curcumin has only 1% bioavailability and a half-life of minutes, 2 or 3 minutes? How can you develop it into a drug? That is the problem with natural products. Now, how to solve this problem? How to just capture the information of a natural product, and how to solve this problem of poor bioavailability? Is there a synthetic equivalent to these scaffolds which are designed by nature or which are developed by nature? One such phenomenon exists in synthetic chemistry or medicinal chemistry is privileged scaffolds. Now, what is a privileged scaffold? Now, this is a scaffold which is drug-like, important thing. And another thing is, these scaffolds are suitable molecular templates for versatile binding properties, which means that these are found in multiple drugs, which means that this scaffold, in this scaffold you can incorporate different pharmacophoric elements to make it interact with multiple targets. Okay, so this is the synthetic equivalent of the scaffold which is found in the nature. Okay, and you see it has the ability to provide effective and selective ligands because they are proven. That's why they are called as privileged scaffolds. Okay, and since it has favorable drug-like properties and it is easily synthesizable and it can be easily modified, unlike natural products, it can be easily modified. Okay, now let us see the example of this, of using nature-inspired design on privileged scaffolds. Why not combine a privileged scaffold with the substituents which is only found in the natural products? Use the same design given by the nature, just change the scaffold. Okay, so if you do this, then you are supposed to get natural product like analogs. It's not a natural product, but it will emulate the information, the chemical information of a natural product. Okay, now let us take and test this hypothesis. Let us take 3 privileged scaffolds. For example, benzimidazole, which resembles purine, and quinoline, which is found in multiple systems, and it is also a plant metabolite, and also Indore. And incorporate these elements. What are these elements? Substitutions which is found in natural products, and develop 3 different molecules. And another important thing to do is to incorporate the hydrogen bond donor combination. That is also very, very important. For example, in the first molecule, you can see this is at the center of the molecule I have incorporated a hydrogen bond donor and a hydrogen bond acceptor. Similarly, in the second molecule, I have incorporated a hydrogen bond donor and a hydrogen bond acceptor. And similarly, in the third molecule, I have incorporated hydrogen bond donor and hydrogen bond acceptor. And this is how we can synthesize. This first molecule is very easy to synthesize by a one-step synthesis, wherein you just condense an aldehyde and not oxidize to give you 2-phenyl benzimidazole. In the second series, you have to synthesize a 2-chloro-3-pharmaquinoline using a Wills-Murray-Hack reaction, and then you condense it with a ketone using non-Angel condensation, and then you convert the 2-chloro to a ketone to introduce that hydrogen bond donor acceptor. Okay, now these are the molecules. Just see. I have taken a privileged scaffold and I have incorporated all the substituents. For example, you can see the presence of a dihydroxy group, methoxyhydroxyl group, and even the chloro group is supposedly a natural product functionality. Okay, so I have incorporated all these elements in my molecules. The only difference is the scaffold. The scaffold is a privileged scaffold. Okay, so I have developed 2 series. Now, if it is a multi-targeted compound, it should show all the properties similar to that of a multi-targeted compound. It should show broad spectrum anti-cancer property, it should be selective, and it should act on 1 or more proteins. Okay, now let us see what happens. So, what I have done is I have synthesized these molecules and I have tested it against multiple cancer cell lines. Okay, for example, breast cancer cell line, cervical cancer cell line, and also lung cancer cell line, melanoma cell line. what I found is, I found these two molecules to be very prominent. They are prominent, why? Because they show broad spectrum anti-cancer property, and it acts on one or more targets. It acts on tubulin, and it also modulates some other targets. Okay. And other than that, these compounds also showed in vivo efficacy and metabolic stability. Okay. Now let us see the example of the first one. Now this work is carried out in collaboration with one of the scientists, Dr. Hoin Lee in Canada. Now these are the molecules, Cancer Therapeutic Research Initiative. Okay. Now you can see how these are the first two cell lines are the normal cell lines. Now look at the other cancer cell lines. Look at the degree of selectivity and look at the efficacy. All are active at sub-micromolar concentration, less than 1 micromolar concentration. Both the molecules. And they preferentially kill a wide range of malignant cells, wide range of cancer cells, breast cancer, leukemia, and also cervical cancer. So many cancer cells are killed, but not the normal cells. There is at least a degree of selectivity against cancer cell over normal cells. Okay. Now, and you can see that they can bypass resistance. Now, this is a problem with as mono-targeted drug or single target therapy, the development of resistance. Now, you can see that it can effectively kill cancer cells which express multi-drug resistance protein and also MRP1, and it can also reverse the resistance of paclitaxel. It was found effective in paclitaxel resistant cancer cell lines. Okay, and other than that, it was found to inhibit microtubule polymerization, just like curcumin, natural products do inhibit tubulin. Okay, and it also showed cell cycle arrest at the mitotic phase. Okay, especially in the SAT checkpoint. And if you look at, it also showed in vivo anti-cancer efficacy in xenograft model for triple negative breast cancer. Okay, now these are the achievements. Now, we got this molecule which was showing a promising potency, less than 1 micromolar potency. Now we went on optimizing the features. We just dabbled with the structure. And what we found? We found some molecules which are so potent that it is showing 6 nanomolar, 3 nanomolar potency against cancer cell lines. Okay, we could achieve the selectivity also. And we also tested these molecules for metabolism, and we could find out that these are much more stable than the previous lead compounds. So, without focusing on a target, we could optimize the biology rather than the binding affinity. Okay, and we had patented this molecule. Okay, and we have published 2 articles which speaks about both the discovery and the lead optimization. Now the second molecule was also found effective in AMRC Canada, but since Dr. Lee was more focused towards the first molecule, he asked us to, he was not interested in taking over this project. So what we did is we found another collaborator in University of Malaya, Dr. Mohammad Rais Mustafa, and we gave this compound to him. Okay. Now what he found is, he also saw the same properties which we found with CTRA, but a little bit less than what we found with CTRA molecules. Now look at these molecules. They also show broad spectrum anti-cancer profile, and they also inhibited polymerization, and they also were effective in resistant cell lines, and they were able to even synergize with some of the clinically approved molecules like doxorubicin, paclitaxel. Okay, and also they were also found effective in human in vivo models, for example, human breast tumor xenograft and orthotopic hepatic cellular carcinoma model. Okay, now this is the first one which talks about the effects of MBIC in breast cancer, where we found that it was showing more than 200-fold potency towards cancer cell rather than a normal cell. And another thing is, you can see the effect, it was showing as good as an effect as clinically approved inhibitors. And another thing is, the same thing, it caused mitotic cell arrest, and also it was found to be effective in vivo. And the same certain phenomenon was found in case of cervical cancer also. Same mitotic arrest, and also it was able to disrupt the tubulin assembly, and also it was able to activate the apoptosis. Okay, and you can see that This paper was published in Journal of Experimental and Clinical Cancer Research. The third one speaks about liver cancer, and in this case, the only difference is we used an orthotopic model and we found it effective. And you can see the degree of selectivity for the cancer cell lines over normal liver cancer cells. The only thing is it didn't work very well with the metastatic version of the liver cancer, hepatocellular carcinoma. Okay, and other than that, it also showed good efficacy in vivo models. Now, together, what we can say safely is natural products can serve us. In the way you look, can serve as a template for designing multi-targeting ligands. I have designed multi-targeting ligands without focusing on the target rather than ligand. So it is a sort of ligand-based drug design, and combining privileged scaffold with natural product functionalities can incorporate natural product likeness in the molecules and can provide molecules with multi-targeting properties. And the third one is CTRI and MBIZ analogs represents prototypes. We are still working on these molecules in our lab, and we have come up with more molecules and more stable versions and more potent versions of it. And they can be a new class of cell line. And you see the simplicity of the design. I have never used any in silico tools for this research. All I said is I just looked carefully at a molecule which was showing multi-targeting property. So with this, I come to end of my talk, and I'm happy to answer any questions if you have. Thank you very much.
Thank you, Dr. Kartikeyan, for your wonderful and very informative lecture. Now, session is open for any question answer.
That is a wonderful presentation, Dr. Kartikeyan, with excellent content and excellent speech. Lot of ideas. What are your strategies for delivering the molecules in vivo? How do you deliver those molecules to the target sites?
Sir, right now we have used intraperitoneal injection. We have not used any delivery system right now because it was in the initial stage. So if at all, if you have any ideas for delivery of these molecules, we will be happy to hear and collaborate, sir.
I guess my question is IP injections probably to the local site where you do you mean intratumoral injection?
Not intratumoral, sir, intraperitoneal.
Intraperitoneal, yes. But the tumor is where?
Tumor is in, for example, in case of breast cancer, it was the xenograft. The tumor is formed in the memory fat pad of mice.
So the
athymic nude mice.
The tumor is in the remote site, not
yes. Not in, no.
That's
yes.
all my question. Yeah. Thank you.
Thank you, sir.
Thank you. Any more questions, please?
Yes, please.
Very, very nice. And I really appreciated your overall critique of our failure to develop drugs in large part, and probably the reason for that we're focusing on a single target, whereas multiple targets were probably what was needed. Now, when in your introduction, you spoke about two strategies. So one was to identify a single molecule that could bind to multiple targets. The other one would be to identify multiple substances, which may actually be in a lot of respects more like naturally occurring plant extracts or something like that. Can you comment on whether you think there could be a strategy for identifying that sort of medication where you actually have combinations of chemicals that produce the beneficial outcome?
Yes, sir. Combination therapy, combination regimens have been known to work. The problem with combination is the dose limiting toxicity, especially with chemotherapeutics. So what I have did is, instead of focusing on the disease system, I have designed a probe and I am questioning the disease system, whether this works with you or not. Because I am not looking at which pathway I have to target. I have just designed ligands and I am using it as a probe. I'm just looking to achieve a sort of domino effect. Maybe I'm lucky, but I'm happy to be lucky.
Thank you, sir. thank you so much for your words of wisdom. It will be a great pleasure for us to listen to you again in our future events. Now, I would request Ms. Shraddha Borwal to present the memento to Dr. Rajesh Sharma, the session chair, and Dr. C. Kartikeyan, the keynote speaker.